Hi, everybody, and welcome to our lecture today, where we are focusing on changing alleles frequency. This is the continuation of our previous lecture, which we focused on constant allele frequency with and the usage of Hartley-Weinberg equation to figure out what is the chance of each of the allele being represented in the population. We also learned some vocabulary that's related to allele frequency. So we are going to expand on that, even though I have provided you with some examples or definitions before we're going to go into it a little bit more, and also look at one of the main concepts, which is artificial selections in so artificial selection as well as positive and negative examples of mutations. So we talked about this in our previous lecture, where we talked about Hardy-Weinberg equation, or sorry, equilibrium, I should say, which was the equation P squared plus 2PQ plus Q squared is equal to 1, and also P plus Q is equal to 1. That the P square is representation of the genotype homozygous dominant. Q is another homozygous, but it's the recessive version, and then PQ is a representation of heterozygous phenotype. We can basically figure out what is the percentage of the population that will carry a specific genotype based on either the parental side or what is the frequency of them being a carrier, or have a specific medical condition related to autosomal recessive disease. Now, we also, as part of the lecture, we talked about 5 major conditions that, if applied to a population, can cause change in the allele frequency and therefore change in a genotype and phenotype frequency. And they were non-random mating, migrations, genetic drift, mutation, and natural selection. So as I mentioned, we did talk about some of these terminology in terms of definition. Well, we will expand on this today by looking at some of the examples in the human population. So this is a picture that represents what those allele frequency, how those allele frequency may change if one of those 5 changes takes place. So on the top, you can see that you have a circle with 4 quadrants, an even distribution of the 4 population represented by their 4 colors. The first one represents the non-random mating, and in this case, we are looking at the blue being a favorite in terms of mating, so it's going to be the most popular one, causing the blue population to increase and the other 3 populations to decrease over time. The second circle at the bottom represents the pattern of migration. So in this case, red and yellow join the group at a higher percentage compared to the blue and green, which causes an increase in their population overall of the total population, and a decrease in the allele frequency for the blue and green. The third one is genetic drift, which I'm going to expand more on it. This is relying on basically random chance to cause and affect the population. In this case, the upper hand was given to green and yellow, causing those populations to thrive in that environment and increase the reproduction. Therefore, the allele frequency has increased. Mutation, again, these can be different types of mutation, but these mutations have favored the blue one becoming red. So there is an increase in the white population compared to the other population. And then lastly, natural selection, which is another part we'll talk about. In this population, gene was selected against, so it was removed from the population. They were not able to reproduce, and you can see we're down to 3 colors instead of the original 4 population or 4 colored population. Now, as I mentioned, our focus on this lecture is how each of these factors are going to affect our overall allele frequency within a population. So let's take a look at some of these factors. So random mating is one of the things that we talked about. And non-random mating basically implies that you're relying on a specific characteristics or a specific situation to put a specific sex at a higher rate of reproduction. And examples of this is observed in all over the world, with the example of Hopi Indians, where you have a characteristic of albinism being the more popular one. We have Genghis Khan from Asia, where there is about 16 million men that are living right now share his Y chromosome, because again he was such a powerful man. So he was selected to reproduce and therefore pass its genetic components and specific alleles to the future population. Certain disease and certain situation, cultural and environmental, has affect a group of population to focus on more of an interbreeding. This is related to disease, which led to development of increased rate of allele frequency to Tay-Sachs disease, as either the reason for that increase was either as a result of very limited population or community where you can marry into, causing endogamy, or marrying your blood relatives that are really close to you, which means they share a lot of that genetic components with you. So please do keep in mind that when you do have conditions like that, where you have certain medical conditions that are more common in the population, if you continue to do interbreeding within that population or within a small portion of that population, there will be an increase of percentage of people that will carry those medical conditions. And again, in cancers, Ashkenazi Jews, one of the medical conditions that have significantly increased over years would be the Tay-Sachs disease. Sorry about that. Now, the second factor we have talked about that will affect the allele frequency is migrations. So migrations is basically where you have within a population, you have pockets of different peoples, and these groups of people are basically defying the Hardy-Weinberg equilibrium by their very existence. So they do not follow that rule, because again, you will have pockets of ethnicity rather than kind of those randomized processes that allows you to use the Hardy-Weinberg equation to solve for genetic allele frequency. So you can have individuals that migrate and move genes from one area to another. So basically, you're bringing a specific allele into that environment. Therefore, the frequency of that allele will increase. In some cases, you also may have people moving, removing certain alleles or move out of a specific area and causing a frequency of a specific allele to decrease from the original population. So you will have an increase in the new population for that specific allele and a decrease in the original population because of the migration patterns. Now, migration can also result in what we call admixtures. Is basically where you introduce a brand new gene to a population, from one population to another population. This gene eventually be passed on to the future offsprings because the one that have migrated into the community now are going to interbreed with the residents of the original population, and causing that gene to be passed forward to the future generations. Now, genetic effects of migrations are also reflected in the current population. As we can see that we have a situation that we use the word climb for it, which is where you literally have the frequency of the allele change in the specific population as these migration patterns take place. Again, you will have introduction of the new alleles into the environment, and when you move out of that environment, so you're using the word immigrant, you remove certain alleles that basically move away from that population. Now, this typically happens, it's not a very fast change. It does happen over a long period of time. It's much more graduate, and it highly depends on the migration patterns and if that migration patterns is continuous. Something some of the barriers that may prevent this process to take place or introducing new alleles is the changes that might be present as a result of language or geographic barriers that limit the access of one population to be breeding with a population in a different area or different environment. Now, the third thing that we're going to talk about in terms of effect on allele frequency is what we call genetic drift. And again, I talked about this before. We're just kind of looking at it in a little bit more depth and looking at some of the population. So genetic drift is referring to the changes or fluctuation in the frequency of your alleles from generation to generation by random chance and changes that takes place in the gametes. It is very specific to populations and is present in all populations. So frequency and the changes or the frequency and allele changes literally causes a genetic drift from one specific allele frequency to another. It can be random or as a result, because it's random, it can be unpredictable. So we cannot figure out what is exactly happening because that genetic drift is taking place. So this component of changing or fluctuation in the allele frequency can speed up when you have a population that the size have significantly decreased in a short period of time. So you're very limited in terms of allele and the frequency that allele have. And then the sampling also changes allele frequency. So if you have a small population or small sample size being removed from that population, again, that can affect what allele has been passed forward and therefore allele frequency over time. Now, components that does affect genetic drift and more specifically enhancing those genetic drift are something we already talked about. Migration is a really big change. Behavior in human, again, preferences in terms of mating. And then the more important one that we as humans typically have no control over is the natural disasters, any type of geographical barriers that causes a small group of population to be limited and disconnected from the rest of the population. Now, as I mentioned, there are things that can cause a speeding of that genetic drift. Two main ones that I'm going to talk about is the founder effect and then population bottleneck, which is kind of what we just described on this slide. Sampling changes as well as the population becoming very small. The proper term again would be founder effect and population bottleneck. So let's talk about this and then I'll So if you look at the word founders as in when you have, the founders of our country, US, right? They're basically where you have a small group from the original population move to a new environment and basically settle in that environment. Now, again, when think about US, the original way that it was formed, we're not talking about the natives that used to live here because that's their environment. We're talking about people that came into the country to colonize the country. Those are the founders of the, or they're the ones that are going to be affected by the founder effect. So the new colony that is coming into that new environment is going to have a very a specific allele that might be different from the original population that came from. So for instance, if you have all of them being of the Scottish background, they may have a higher allele frequency depending on the environment, versus if the original colony came from England, they may have different allele frequencies depending on the environment. So whoever comes in, they will have very specific allele frequency that is related to their origins, the original population they came from. Now, we do recognize that the founder's effect is in a community, as well as our local history has been established. There are examples of this where a specific trait has been passed down from one generation to another generation after the migration has take place, or the movement of the population take place. And some of these founder effects have led into a specific medical conditions or illnesses that have been carried to the population, where if you look at the original population, that number would be significantly lower. But because you have these isolated colonies as a result of the founder effect, the effect of that medical condition would be more significantly visualized compared to the original population. Here is some of the examples of the founder effect. So you have the multiple population examples. You're welcome to look through them. We just want to kind of look at some of the examples of this. We have the Costa Rica population, where the original people that migrated were about 4,000 people. And within 12 generations, you end up about 2.5 million people. And then another example, you have people from the Iceland, where the original population that migrated was about 25,000. And within 40 generations, you're ending up with 300,000 population. Quebec is another good example. Quebec, Canada, the original population that migrated were about 2,500. And between 12 to 16 generation, your number has gone up to 6 million people. So again, this is the original population, right, where they came from, and this is the final population that is created based on their new environment. Now, because the original genes from these numbers, this last column, comes from only these people, like as low as 80 people, guys. So you have very low number of frequency of allele frequency compared to the original population that is depicted here, because you're taking a small sample of that population and taking their alleles to recreate the next or create the next generations to come. Now, here's a couple of examples of founder's effect in terms of medical conditions. You have Afrikaners of the South Africa, more susceptible to porphyria variegata. Sorry about if I mispronounce this. Another thing that you want to kind of notice this is evidence that where you look at the original population, again, the certain mutation or certain medical conditions have a much lower frequency versus a different population. So for instance, people in Puerto Rican ancestry in East Harlem have a higher chance of getting the Steele syndrome. Now, if you're looking at these individuals in these populations, the number of individuals that may be affected by these medical conditions and illnesses may be significantly higher compared to where that population actually came from because of the founder effect. So going back to the picture that I had earlier, here is the founder effect. If this was your original population, you can see a significant diversity. And as you move forward, we have taken this sample, and this sample has moved into a new environment to form their colony. And this colony is now have the frequency of this population. And you can notice that the blue and the red are significantly less frequent compared to the other color. And then the yellow and green are at a higher frequency because of the original sample size that we have taken. The bottleneck is another concept that, again, leads to the concept of genetic drift. As the word bottleneck implies, you kind of have that narrowing of the population movement from one area, or sorry, the narrowing of the population passing certain characteristics to their offspring, depending on their environment and the changes in the environment. So in the case of population bottleneck, we're usually talking about some sort of significant change that destroys or damages most of the population, and therefore only a few a specific individual with specific alleles are left to create the next generation and offsprings. Therefore, the allele frequency are being replenished from a very small portion or sample size of the original population. In this case, again, your gene pool are much smaller compared to the original ancestral gene, where you had a huge number of species where they could have basically interbreed with each other. Now you have a much smaller size with very specific allele frequency that might be either higher or lower than the original population. Now, again, population bottleneck can happen if you have things like natural disasters, or when you have people being separated from from the original population by geographical barriers, specific animals. Again, for instance, if somebody comes and colonize an island. Here's a depiction of what bottleneck looks like. Again, if you have equal frequency of all genetic alleles in the bottle, that bottleneck may allow, let's say, the green to be more frequent as a result. And again, it's completely by chance. And that causes the frequency of the green to increase over time compared to red and blue, for instance. Some of the examples of the population bottleneck is found in one of the Hawaiian Islands. People that are associated with people of Micronesia. This is a group of people that, again, live in an island. They're very highly isolated as a result of a typhoon that created the original bottleneck. Now, this population, because of the allele that was selected as a result of this natural disaster, are much more susceptible to blindness, and a condition called acrometaplegia is a very specific form of color blindness. So they're either born blind or they have very limited capacity to distinguish between colors. If you look at the population of the world expanding from its origin, which is Africa, if you look at the diversity that's observed in Africa, in the original population in Africa, it's much higher. It's the genetic is very diverse, but people who migrated out of that environment had led to a very specific allele being moved away from Africa, and therefore they have a significantly less diverse allele frequency compared to the original of people that live in Africa. And then Ashkenazi Jews is another thing that we already mentioned before. As a result of the environmental changes, cultural changes, massacres, preference in terms of mating has contributed to major medical conditions among the Ashkenazi Jews. And again, it can be related also to the fact that they have a very small community and they do a lot of interbreeding, as well as interbreeding within family, close relatives, like first cousins. Here is some of the medical conditions that have been more prevalent among these Ashkenazi Jews. And again, you notice that there are many different ones, and they are very high frequency in terms of being carriers of this medical condition. So notice we're talking about PQ. So this is a heterozygous genetic. We're not talking about people actually having the medical condition, rather they being carriers and therefore having a higher chance of passing that to the kids. Things like Bloom syndrome that causes significant decline on terms of immunity, sensitivity to light, sorry, fertility levels being lower, breast cancer associated with BRCA1 and BRCA2 gene. Again, 3 out of 100, which is basically 1 out of 33. Um, Another one that you might have heard of about is Tay-Sachs disease, which we have talked about multiple times already. Many, 1 out of 26 exons genes are identified as carriers, which is a very bad in terms of affecting the nervous system, and the chance of survival past the age of 4 is very rare. Now you're welcome to look through the other disease like Gaucher disease, the different type of anemia, Canavan disease, where again a lot of them are associated with either blood or nervous system damage. But regardless, our goal is to look at how frequent it is for these individuals living in these populations to be the carriers of these very rare medical conditions that have a significantly less frequency in the general population. But when it's compared to Ashkenazi Jews, they have a very high probability of being the carrier of these medical conditions, and therefore having a higher chance of children being born with these medical conditions. Now, mutations, again, we have talked about it in details in full lecture, so I'm not going to expand on what is mutations, but anything that's related to the change into the original DNA sequence will be considered a mutation. In some cases, the mutation can cause one allele to change to another, and therefore change the frequency of alleles over time. Now, these variations in the genes may arise from things like crossing over and independent assortment, which again, we talked about it in Mendelian genetics. If you don't recall it, please go back and review it. In terms of mutations, I think this is one of the things that I talk about, which I think it's very fascinating. So when we talk about mutation, the concept of natural selection, so choosing a specific characteristic and choosing for a specific characteristic to increase the chance of survival for that individual. So natural selection basically allows you to eliminate alleles that may negatively affect you, but the recessive alleles will be maintained in terms of the heterozygous, right? So that means that your Q2 might be very low, but you still may have a chance of carrying it in form of the heterozygous genetic information. Now, genetic load is another word that you guys want to kind of hear about. Is a collection of recessive deleterious alleles in the populations. This genetic load, this concept of genetic load can be influenced by things like human behavior and events that have happened in the previous generation. For instance, one of the medical condition called phenylketonuria, which is what I mentioned before, PKU, inability to break down a specific amino acid. The mutation originally was very small in terms of how it impacted the individual and the population. For most of the time, it was a silent mutation, which means it didn't really cause any changes in terms of the protein. However, when it is kind of coupling with other environmental factors, such as very small population samples, because you have been separated from the original population, or a specific type of breeding, like the non-random breeding that I mentioned, these mutations may have a significant effect on the allele frequency and the population overall. For instance, the deletion mutation that is formed in the originally Israeli Yemeni Jewish, which originated from the middle point right here. It originally, well, it is believed, based on the history and looking at the genetic information, it starts from the Sana'a area in Yemen in around 1850s. Sorry, 1750s, not 1850s, 1750s. And these alleles spread either northward, going upward towards Saudi Arabia, uh, and these are represented by the solid arrows, by the way. So these are the arrows you see right here. And then subsequently, as time passes by, they have spread, eventually going more southward and westward, and eventually reaching Israel, affecting the population of the Jews in Israel. So the importance of this, again, is why are we looking at this, is that the mutation actually started from a very specific area, right, where you have a large population. But as people migrated and moved away from this area, and the sample size of the population became smaller and smaller, then they carry that medical condition to a new environment with a smaller sample size, which means the allele frequency for having these medical conditions have increased over time. Now, natural selection, we have talked about it again. I think it's cool for you guys to understand that, as I mentioned, yes, there are factors that the environment select against, like what we talked about in terms of medical conditions, right? You selecting against it because you don't want it passed on to the next generation. But sometimes the natural selection also chooses for things that will benefit us. For instance, the concept of lactose tolerance is our ability to maintain a gene that help us break down the dairy product, the sugar in the dairy products. So again, natural selection selected for that, allowing us to keep that characteristic and basically be able to consume dairy product regardless of the type of dairy product. So it's a tolerance. And if you have the recessive version of this, it causes lactose intolerance. Now, another concept that we want to kind of talk about briefly, which I already mentioned in the previous slide, again, is the negative selection versus a positive selection. So kind of putting that concept that in natural selection, you can have genes that are selected for because it's benefiting the population versus benefiting the population and allowing the reproductive pattern to continue, reproductive success to continue, versus negative selection of the natural selection is where you have genes that are actually harmful to the population, causing its demise, and therefore the frequency of that gene will be significantly reduced as it doesn't get passed on to the future generations through the process of reproduction. So hopefully all of you have heard of Darwin, who actually introduced the concept of natural selection. He believed that in, well, in his time, the belief was that natural selection is primarily negative. So again, you're choosing to eliminate genes that are going to be harmful to you. But with our ability to sequence the genomes and looking at our comparing to the original population and see how the genes have been maintained over populations, we have figured out that many of the genetic information that is present for us are actually positively selected for, that allows us to again survive in our environment better rather than eliminating genes that can be harmful to us. And as I mentioned, is our ability to tolerate lactose within our body, allowing us to continue consuming dairy product as part of our diet. Another example of the positive selection is related to the Sherpas that live in the um, Himalayas. Um, so they're the ones that basically help with individuals who want to climb Mount Everest. They have a very specific gene called EPAS1 that is selected for. So again, it's a positive selection that allows them, to be very or highly adaptable to an environment with very low levels of oxygen. So if we were to introduce to that environment, we would not have much of a chance surviving. We need oxygen because the amount of oxygen is significantly lower. But in these sharmas, because of these uh, positive selection, they have the capacity to adapt at very low environments of oxygen, allowing them to climb Mount Everest multiple times during their lifetime. Now, everything we have talked about so far is related to what naturally happens in the environment. It's not something as humans we control. However, that being said, there are a lot of factors and components that we do control through the process of artificial selection, where we choose a specific traits and we continue to breed selecting for those specific characteristics. This happens a lot in our plants, and it also unfortunately has been happening in the most recent decades in our domesticated animals. So we're choosing for a very particular phenotype. Um, for instance, here is a picture of a Great Dane with a Chihuahua. Both of these come from the same species. However, because of artificial selection as well as intense interbreeding, so we're talking about kind of like going family to family to family within the same population. So again, not expanding it into, that randomized concept and introducing the population to the dogs to being introduced to the whole population. That caused a significant increase in our ability to first select for size. So again, you can see that difference. And then the second thing is the concept of domestication. So it's kind of controlling the things that, the genes that control the animal nature of the dogs and taking those out and allowing the animal to be more domesticated. Now, natural selection, even though it's a really good thing for us humans, we're not the only one that can utilize that natural selection for their survival and the purpose of reproduction. So the example of natural selection is also viewed in our environments, with one of the most important and the scary ones being antibiotic resistant infections, where you have mutation in the specific bacteria where they become um, resistant to the typical antibiotics we use to deal with infections. The biggest one that we see nowadays is associated with a specific version of the Staphylococcus aureus, causing um, MRSA. And this is basically the one that is resistance to methicillin, which is basically the um, uh, family of antibiotic like penicillin and ampicillin. Uh, and this is really dangerous for us. To kind of show you what that looks like, here is a group of bacteria we start with, and this is before anything has changed, no mutation, and your antibiotic can kill all of them. Now, as time passes by, generations are created. Your generation two, you will have a mutation in one of them. It's represented with red, and that mutation is basically um, causing the um, or having the capacity to withstand the antibiotic. So when you introduce the antibiotic into this population, right, most of the greens, if not all, are going to die off. And the only one that's going to continue reproducing would be the red ones. So within the next few generations, the red ones are going to basically take over the overall population of the bacteria. And now you're going to end up with huge number of red that are effectively tolerating the antibiotic, therefore becoming antibiotic resistance. So one of the best ways to prevent development of these antibiotic resistant bacteria is as a human, as an individual, is one, do not insist for your doctor to write antibiotic if you have a flu, because flu is a viral infection and antibiotic does not do anything for you. And second, if you are prescribed antibiotic, make sure to take the whole sequence of that medication, not to miss a dosage. And if it's 5 days or 7 days or 10 days, make sure to complete it, because then you have a higher chance of getting rid of that little red one, which means we are less likely to produce antibiotic resistant bacteria for our future generations. Now, the last thing I want to talk about is, which is, I think, again, really interesting, is when we have mutations which are recessive, so autosomal recessive genes, where if you have two copies of them, that will cause a major medical condition or disease within humans. However, they also provide protection against another disease or another medical condition. And therefore, you will have a lot of heterozygous um, individuals where they have uh, the characteristics of the recessive disease, but they're only carriers. So again, so instead of having QS square, where you have the actual medical conditions, you're going to end up with a higher frequency of the PQ as the heterozygous. And this is again advantageous because it allows you to be only the carrier, so not necessarily being affected by the disease, but also getting protection against a second disease that would normally cause harm to you. So one of the best examples is the sickle cell anemia, which was the medical conditions where your red blood cells are sickle shaped or kind of crescent shaped. Now, due to the fact that if you are a carrier, you may have a much higher resistance to malaria. Malaria is as a result of an insect bite, which can cause major health conditions and ultimately death in many African countries. However, if you're identified as a carrier of this sickle cell gene, you are typically resistant to malaria, or you have a much lower symptom development when you are diagnosed with malaria, which is very good. Now, how does it do that? When you have sickle cell anemia, again, your blood is significantly, not significant, I should say, is thicker than normal. One of the main reasons is because the crescent shape of the um, red blood cells, instead of being round, and this difference in terms of the shape hampers the parasite ability to infect. So your mosquito, even though may bite you, they cannot have the malaria being passed down to you. And again, the bend shape also prevents these parasites from growing and blocking and spreading the infection. So the shape, even though it's not the healthiest version of the cell, it is beneficial to protecting you against a secondary and a very dangerous um, disease that can lead to death in many cases. We have other examples of uh, blood disease, more specifically red blood disease, such as the thalassemia and hemoglobin C, that both of these basically cause damage to the globin protein of the red blood cells. They're not, uh, so again, if you're a carrier for these medical conditions, you are less likely to be um, affected by malaria, or if you do have it, it would be significantly less symptomatic because of the change in the red blood cell protein hemoglobin. Here is some of the examples of what is this kind of the polymorphism looks like. We call that balanced polymorphism, where you have one medical condition, again, causing protection against secondary medical condition when you have the heterozygous version of the gene present. So G6PD is a mutation in the globin protein that again prevents you from developing malaria. Sickle cell has done the same thing, limiting the ability of the parasite to either release the toxin in the first place, or even if they are in your blood, they are unable to infect your red blood cells. Phenylketonuria is another one we talked about, which is basically inability to break down a specific amino acid. However, if you are a heterozygous for or carriers for this medical condition, you can protect the protect well, I should say, the fetus is protected against many of the fungal infection. Prion protein mutation, which is something we talked about before. If you are a carrier, it protects you against transmittable spongiform encephalopathy, which is a brain condition, so again, affecting your nervous system. If you're a carrier of cystic fibrosis, you are less likely to have diarrhea associated with cholera. So to just kind of show you that. So if you have cystic fibrosis, which is again building up that thick mucus in the respiratory system and having effect in immunity, etc., it may protect you against development of disease that cause severe diarrhea, like cholera, which is basically an infection as a result of a bacteria causing severe vomiting and diarrhea. You basically get dehydrated, and that leads to death. So the gene that leads to cystic fibrosis, if you are a carrier for it, it causes sorry, I should say, things like typhoid fever or even cholera requires a functional, which means the CFTR, to be functional. So if you're a carrier, that gene, at least one copy, is not fully functional, which means that the chance of the infection actually taking hold of you and causing diarrhea is significantly less. And the last picture I wanted to kind of show you is the pattern that you observe in terms of the relationship between this concept of polymorphism for malaria and relationship with sickle cell anemia. So the areas where you have typically high levels of sickle cell anemia, sorry, you have area where there is no effect on in terms of the infection. So if you have really high levels, you may actually see a significant decline in terms of development of malaria, which is really good. And then I just want to kind of highlight a couple of other medical conditions. And again, if you have a Smith-Lemli syndrome, you're protected against cardiovascular disease, which is absolutely amazing. And kidney disease as a result of variation in G1 and G2 protein prevents you from African sleeping sickness. This is more common among African populations. So that's about it in terms of our genetic information and variation in changing allele frequency. I do want you guys to please make sure you can provide description for each of the 5 types of factors that affect change in allele frequency in a population. And also, please make sure that you are able to associate each of the examples I provided with one of those 5 components. So if you have an example of, let's say, a medical condition such as cystic fibrosis, can you associate that, I guess associate that with a specific characteristic that may lead to an increase or decrease in a frequency of allele in the population. So that's about it. And thank you for listening. After this, we only have one more lecture. So we will talk about the genetics of cancer, and that will be the last lecture for our course. Thank you, guys.